Our next speaker, is Rich Klinghoffer. He is the CSO of Presage, where they actually have a very powerful platform, allow you to screen multiple drugs directly in tumor in their native microenvironment. And as you would imagine, a microenvironment is such a big player for immunotherapy, so it definitely have great potential. Prior to Presage, Rich is at Merck, which was at Merck, where we overlapped, and I was fortunate to witness firsthand how effective he was as a lead for target validation and identification, and his team actually really filled the pipeline for Merck Oncology. So I'm so glad to be able to see Rich again over 10 years ago, right? So thank you, and look forward to your talk.
Thank you, Teresa. So Presage is a small, so we're about 20 people, a little over 20 people, cancer-focused company. The technology I'm going to talk about today, Cibo, I think intuitively can be used as a companion diagnostic. At Presage, we're not intending to develop it as such. We're actually developing it as a drug development tool, and I'll go over that at the end of the talk. But I do think that in order, what I'd like to hear from many of you in the crowd today after my talk is what you think of the potential for taking it all the way as a companion diagnostic. Presage, as a small organization, it simply does not fit with our business strategy. I think in order to develop it as a diagnostic, it would have to be placed in the hands of someone like Matt, who really has the expertise in taking an assay all the way through FDA approval. So this is a number that's thrown around a lot. It's a sobering number. So 9 out of 10 times a new drug enters the clinic, it doesn't provide any benefit to the patient. And so this is a number that inspires many of us in the cancer drug development industry to find ways to improve this process. And when I was leaving Merck and thinking about what to do next, I was thinking, well, how can I contribute to this? And I figured that folks like Teresa and others had the genomic space pretty well covered. And so at the time, I was thinking about, well, my hypothesis is the tumor microenvironment matters. And when we're developing drugs preclinically, we often fail to really model the complexity of the microenvironment really well. So I think all of us can appreciate that tumors are not simply homogeneous bags of cells. It's widely accepted that there's quite a bit of heterogeneity, not only in terms of the number of cells that can be in a tumor, there's complexity in terms of the interaction between these cells and their extracellular matrix. There's regions of high vascularity and hypoxia. And of course, as we're all well aware, there's quite a bit of interaction, a very important interaction between the immune microenvironment and cancer cells. And so what we were trying to think about is, could we develop a way to truly interrogate cancer cells directly in a native microenvironment, ultimately the patient's own tumor? And could we do it in such a way that we could look at multiple potential therapies simultaneously in the context of that tumor? And so the concept that I was thinking about at the time was similar to a test that's been around in infectious disease for years, the classic Kirby-Bauer antibiotic resistance test, where you have a petri dish with a lawn of bacteria, and you place little wafers that are impregnated with different antibiotics, and those that clear the lawn of bacteria in a spatially defined manner are the ones that are effective, and those that don't are not. And this is a very easy way to figure out which antibiotics to treat that particular infection with. The challenge for us was, could we pull off something similar in the context of a much more complicated system like a solid tumor? And so that's where we come to Cibo. So Cibo stands for Comparative In Vivo Oncology. And this is what you're looking at here is a thin section of a tumor that has been subjected to a Cibo procedure. The blue stain is just a DAPI-stained background, and what you'll see within that DAPI-stained background are these little green dots that represents a fluorescent tracking marker, which I'll go over in a moment, that is inserted along with different drugs at these different sites. And you'll see that some of these sites have this reddish stain around them and some of them don't. That reddish stain is dead cancer cells or cleaved caspase-3 stained cancer cells that upon exposure to the drug underwent apoptosis. So it's a very simple way of looking at which drugs are working and which drugs don't, analogous to the Kirby-Bauer antibiotic resistance test. So the Cibo technology consists on the front end of an intratumoral microinjection device engineered by Alicia Moreno-Gonzalez, who's here in the audience today. It enables the first side-by-side comparison of multiple drugs and drug combinations simultaneously in a single living tumor. We use very small amounts of each drug or drug combination, so it is indeed toxicity sparing to the patient. And we're using this as a way preclinically to improve the probability of success of drug combinations and testing them prior to conducting traditional clinical trials where you obviously expose the patient systemically to drug and therefore all the risks associated with that exposure. Here's the handheld device engineered by Alicia and her team. Just very simply, the device works by depressing this lever, which results in retraction of these needles, which are end-loaded with drug. And so and I'll show you a picture of this in a moment. When the needles are retracted, this leaves behind columns of each drug that are inserted into the tumor. We've completed a first-in-man feasibility study with doctors at the Seattle Cancer Care Alliance and patients with palpable lymphomas. We've performed the procedure on over 70 canine patients with sarcoma and lymphoma to date. We have a sarcoma study that's starting now at the Seattle Cancer Care Alliance. And we're planning on testing the technology with an investigational agent, our own. We have a phase 1 anti-cancer compound, and that's planned for this year. This is some of the data from the study performed at the Seattle Cancer Care Alliance. This is actually the hand of Dr. Oliver Press inserting an early version of the technology directly into a palpable lymph node. In this figure, he inserted vincristine, and the way we quantify the data is using radial distance from the initial site of the injection marked by the fluorescent tracking marker as a surrogate for drug concentration. So each and every injection site results in a drug concentration curve. So the closest to the injection site, so these are microns, results in the highest amount of drug concentration and typically the highest amount of functional response, and the lower concentrations represented by the farther away from the injection site and usually a much lower response. The response dissipates as you get farther and farther away from the injection site. So here is a bit more detail about the Cibo platform. The Cibo platform can essentially be broken down into two pieces. On the front end is the injection device, again inserted here into a sarcomas mass of a canine patient. And on the back end, an image analysis package. So following insertion of the device into the tumor, the injection takes place over the matter of seconds. What it leaves behind are not only the different drugs or drug combinations, but you see here again, right through the skin of this sarcomas mass, is the appearance of the fluorescent tracking marker, which denotes the position of each and every injection site throughout the entire process from injection through analysis. You'll notice that some of these fluorescent tracking markers are different colors. That allows us to clock the position of the tumor throughout the process from injection, again, to analysis. As I mentioned before, following insertion of the drugs, we typically resect the tumor, particularly in the clinical setting, 24 to 72 hours later. And each injection site leaves behind a track of drug due to the way Alicia has designed the device. This is important for two reasons. One, it allows us to section crosswise to the injection column to assess the responses to the individual drug inputs. And it also allows us to sample across the z-axis of the tumor for different microenvironmental effects. This is a gross section from that very same tumor, and you can see the retention of all the fluorescent tracking markers. And then 99% of our analysis is performed by IHC. This is a thin section, again, from this tumor over this blue DAPI stained background. I hope you can see the fluorescent tracking markers. And around 2 of them, you see this region of cell clearing, so this black area within that blue background around positions 2 and 4. And around that black area, a perimeter of red stained cells, again, cleaved caspase 3 positive or apoptotic cells. So again, this provides us with our first sense of which drugs are working upon insertion into that tumor and which drugs are not. And the second component of Cibo is a software package, automated software package called Cibo Analyzer. Cibo Analyzer takes scanned images, identifies each and every injection site by virtue of the fluorescent tracking marker, creates these regions of interest, and then within those regions of interest, analyzes each and every cell down the column for whether they respond to the drug or whether they don't. So the cells that respond, so this is just a magnified image from this injection site or this response site, are denoted here with these green dots. The cells that don't respond are denoted here with the red dots. And sometimes it's these red dot cells, the resistant cells, that are exposed to drug but are not responding, that are the most interesting to try to characterize biologically, because these are the cells that could potentially represent resistant disease, which grows back the tumor following initial remission. And here's an example of that. So in this particular case, we had a drug that was a covalent binder to its target. And we happen to have the luxury of an antibody which specifically saw that drug target conjugate, and that's shown here in yellow. So we could use that to track the biodistribution of the drug. And we asked the question, well, whether this drug induces an anti-cancer response in terms of apoptosis. So we took a serial section and stained it with cleaved caspase 3, and you could see that in the approximate region covered by the drug, we do indeed induce apoptosis. But there are many non-cleaved caspase 3 stained cells left behind, and these are indeed live cells, so we asked the question, why? And so our first hypothesis was that we were just getting incomplete target engagement. So we stained for the target, shown here in pink, and we could see that within the region that was covered by the drug, we actually do get pretty good homogeneous target knockdown. So that was not the answer. So we started to look at various potential microenvironmental effects on cancer drug efficacy. And one of the ones that we looked at was a marker for hypoxia, and that's going to be shown here next in cyan. And you could see that in a good portion of the regions that were devoid of cleaved caspase 3 positive cells, these are now filled in nicely with these regions of hypoxia. We know this is true hypoxia in that not only does it express the right biomarker, but when we stain for CD31, we see an inverse relationship for vascularity as seen by CD31 and hypoxia. And one thing I also want to point out is that, again, as I mentioned before, the tumor cells that are exposed to the highest level of drug are right next to the injection site. And what you'll notice, and what I didn't notice until we did this stain, was that there were a number of cells that were not cleaved caspase 3 positive that were living right at the highest levels of drug exposure. And so what this suggests to us is that this drug, while effective in some portions of the tumor, will never be fully effective at even at the highest doses of exposure, and therefore requires combination regimens in order to be a truly effective drug. So these are the types of studies that drive our drug development hypotheses or drug combination hypotheses. Just want to quickly go over a fundamental experiment that we performed in the very early days of Presage to address the question, do the localized responses that we see following introduction of the drugs to Cibo equate to or predict out systemic responses, at least in the context of the preclinical setting or in mice. So in this case, we took drugs that were commonly used in the lymphoma clinic, doxorubicin, vincristine, and mifosfamide, which is the active version of the prodrug more commonly used cyclophosphamide, inserted them into 2 models of lymphoma, one which we call chemo-naive, and one a version of this which was rendered in the lab to be resistant to doxorubicin by a step-up procedure. And what we saw was that when we exposed the chemo-naive version to doxorubicin, it induced DNA damage by virtue of gamma H2AX stain. and some localized cleaved caspase 3 in the chemo-exposed. At the same concentration shown here, there was no response. Vincristine induced its expected response. Mitotic arrest is seen by phosphohistone H3 stain, and again, a corresponding region of apoptotic cells. Again, no response in the chemo-exposed version. The cross resistance is likely due to PGP pump activation, so we did perform molecular profiling with Covance, and they saw that there was high levels of PGP pump, so that probably explains this resistance here. With mifosfamide, it was more interesting than both of these in that not only did mifosfamide retain its ability to induce DNA damage, but you actually saw an increase in cleaved caspase 3 positive cells. So we were interested to see what would happen when we looked at these drugs following systemic delivery. Again, the chemo-naive and chemo-exposed models. In the chemo-naive model, all the drugs had an effect, but in chemo, that Cibo predicted out in both directions, heightened sensitivity and resistance. And this was published in our paper last year. So as we wanted to move into the clinic, and we have, as I showed you before, with the early patients that were subjected to the technology by Dr. Press, we do face, as you might expect, a number of regulatory hurdles to pursue clinical trials in patients. As a surrogate to really learn more about our technology in the context of a clinic-like setting, we have learned quite a bit from a very valuable collaboration with a number of Seattle area vet oncology clinics, which collaborate with us under the banner of Woof for the Cure, working on our fight for the cure. We have 8 sites now and 10 oncologists working with us. And so what this gives us an opportunity to do is really assess microenvironmental effects to each of these, to the inserted drugs or drug combinations with an immune component that we can't get from our typical immune deficient mice. And so here again is a soft tissue sarcoma, a block. You can see all 8 injection sites by virtue of the fluorescent tracking marker. And at 2 of these sites, you see nice overt cell clearance. Both of these sites contain doxorubicin. And at the other sites, you do not see this overt clearance. We did use other agents than doxorubicin that are commonly used in the soft tissue sarcoma clinic, including gemcitabine and docetaxel. Similar to historic single-agent responses to gemcitabine, we really never see much of a single-agent response. With docetaxel, we always see a response in terms of mitotic arrest in these abnormal mitotic spindles. But really, only doxorubicin, which has been the mainstay of soft tissue sarcoma therapy for now several decades, is the agent that induces this overt cell clearance. So one of the reasons that this may be the most effective agent to date is that, unlike the other agents, doxorubicin not only induces a cancer cell autonomous effect, but also is able to bring in cytotoxic T lymphocytes to mount an effect, to mount a concerted attack on the cancer cells. So you see that when we expose this portion of the tumor to docetaxel, you see no cytotoxic T lymphocytes or CD3 positive cells, and that's our best biomarker to date in the canine setting. But around this region of cell clearance induced by doxorubicin, you see around the perimeter lots of CD3 positive cells. Our best evidence that these are indeed active cytotoxic T lymphocytes is that when we take a magnified image and look at these cells and co-stain for not only CD3 but along, with granzyme B, we can see that these cells are expelling their payload or their cytotoxic payload, this granzyme B, into adjacent sarcoma cells. Not every patient in the human clinic responds to doxorubicin, and not every patient subjected to the Cibo procedure with doxorubicin showed a response, and this is depicted here. So in this experiment, each of these spots represents an individual canine patient. And what we did here is we used gamma H2AX, or DNA damage, as a surrogate for drug distribution of doxorubicin, and that's plotted here on the x-axis. and loss of vimentin-stained cells, so sarcoma cells stained positive with vimentin as a marker for cancer cell death. And when we used k-means clustering, we were able to identify 3 groups from this plot. Responders, as depicted up here, which showed good distribution of doxorubicin and good cell clearance. What we call structural non-responders, tumors that showed poor distribution of dox and therefore not surprisingly poor response. And then what we call molecular non-responders, or situations that are what we typically think of when we think of drug resistance, where we get good exposure to doxorubicin, but poor response. And one thing I want to point out is that whereas I think everyone intuitively appreciates that tumor architecture or drug distribution matters in terms of response, we rarely look at this when we talk about precision oncology. And here I just want to point out that when we did stain these sections with a Mason trichrome stain, you can clearly see the difference between our structural non-responder, which has a very packed cell density with, I hope you could see this here in this slide, thickened collagenous stromal matrix, that bluish area there, versus the responders and the molecular non-responders. So we then asked the question, well, could we reverse any of these non-responders with combination therapy? And importantly here, we did a series of experiments in those very same dogs with a preclinical autophagy inhibitor. Now, what I want to point out about this autophagy inhibitor, which we call PS1001, is that this is a compound that should be nowhere near a large animal. It's toxic. It has very poor PK, thus very difficult to deliver in an in vivo setting. But by virtue of using very minute amounts of this with Cibo, we're able to assess just the fundamental biological question: does autophagy inhibition reverse the response to any of the doxorubicin non-responders? We do know that doxorubicin exposure induces autophagy, and that autophagy is a common mechanism of resistance to many different anti-cancer agents, including anthracyclines. And this slide is just to point out that in the region exposed to PS1001, you do see this classic hallmark of autophagy inhibition, this punctate LC3B stain showing these immature autophagosomes that are unable to fuse with the lysosome. And so, as I mentioned before, we subjected the same set of dogs to not only doxorubicin, but at a different site, doxorubicin plus PS1001. And what we found is that in the structural non-responders, PS1001 had no effect. Again, drug can't distribute, and PS1001, or inhibition of autophagy, doesn't loosen up the stroma in any way to allow for distribution. But in 3 of the 5 molecular non-responders, we do see an increased response that is essentially equivalent to that of our responder group. So not every patient responds to combination therapy, but an encouraging 3 out of 5 did. And so I just, again, want to point out that one thing that that Cibo enables you to do, which we typically don't do in the preclinical setting very well, is sample a diverse patient population and really assess how your drug or drug combination does across that patient population. So whereas our initial hypothesis was that autophagy inhibition would act in a cell-autonomous way with in concert with doxorubicin to kill the cancer cells, we learned a number of things about doxorubicin, or about PS1001 in the following experiments, suggesting that it may have an important impact on the tumor microenvironment as well. So in this figure, we were staining for macrophage infiltration. And if you compare one of these converted non-responders to compare doxorubicin or doxorubicin plus PS1001 to one of the converted non-responders, you see this increase in MAC387 stain, so an increased macrophage infiltration. Initially, I thought that that might just be because now we do have a region of cell death and the macrophages were simply coming in to clean up that debris. However, we also saw that an increased infiltration of macrophages in our responders, which have essentially equivalent levels of cell death in the absence or presence of PS1001. So it seemed like this autophagy inhibitor in both of these contexts was increasing macrophage infiltration. So what? Well, macrophages can have two very extremely different functions depending on their polarization state. There are two extreme polarization states in macrophages. M1 polarization state, which is an anti-tumor polarization state where it mounts an anti-tumor effect, or an M2 polarization state where it actually has a pro-tumorigenic or pro-tumor survival effect. So we wanted to define whether this influx of macrophages was M1 polarized, anti-tumor, or M2. And we did it with the following stains. So a marker for M1 is phosphostat1, and a marker for M2 is a CMAF. So we co-stained for not only the macrophages, but the polarization marker, and saw a nice overlap between macrophage and phosphostat1, or the M1 anti-tumorigenic stain, but no overlap between the macrophages and this M2 CMAF stain. And so what we believe PS1001 is doing is not only increasing recruitment of macrophages to these areas of autophagy inhibition, but skewing their polarization state to M1. Is this talk getting too sciencey? You like this? And then just to bring it all together, here's another section from one of these, what I call converted non-responders, where indeed around the fluorescent tracking marker, you see this nice perimeter of LC3 positive cells showing autophagy inhibition. Within that region, you see this influx of M1 anti-tumorigenic polarized macrophages, so co-stained for MAC37 and phosphostat1. We also noticed that there were a number of MAC387 negative phosphostat1 cells within this region of infiltrating T lymphocytes. So we believe that not only is the autophagy inhibitor preventing autophagy in the cancer cells and thus helping doxorubicin kill them, but also helping mount this immune attack on the cancer cells as well. And so currently we're working with this hypothesis or this model that these M1 polarized macrophages, which are phosphostat1 positive, are secreting cytokines that are inducing phosphostat1 signaling in lymphocytes, recruiting them, and activating them so that they can also mount a response to the cancer cells along with doxorubicin. Okay, Cibo Clinical. Alicia and her team, again, are leading this effect. What we want to do is, in terms of indications, is first establish whether Cibo not only can predict the outcome in terms of preclinical experiments but also whether the localized responses induced by Cibo can predict patient outcome. This is the fundamental question which we have not addressed to date, but that our next sarcoma trial is designed to address. The disease indications we're going into are ones that are superficially located tumors. This is the logical place for us to start. I'm not worried at this point in time about deeper set tumors. If we establish the fundamental hypothesis is true that the localized responses will indeed predict out patient response, then I think it's worth to invest in further engineering so that we can get to deeper set tumors. But until that is addressed, I think that remains debatable. So these are the indications we're going after. This is essentially the trial that we're performing at the Seattle Cancer Care Alliance with Cheppy Conrad and Seth Pollack. It's a small trial, 12 patients, where we'll compare localized response to patient outcome. And these are some of the drugs on our list that were approved to use in this trial. In terms of our regulatory status, we're able to perform these trials under non-significant risk designation that was given to us by the FDA back in March. What this allows us to do is perform our studies following IRB approval, which we have received, with FDA-approved agents only. We can perform experiments with investigational or non-approved agents that have an open IND, but you have to add on our protocol to an open IND in order to pursue that, and that has to be further reviewed by IRB. As I said before, we're not pursuing Cibo as a diagnostic or companion diagnostic. I think that would, again, have to be done in the hands of a much larger organization than Presage. So we're developing it as a drug development tool. And so we're on the path to do so, and what qualification as a DDT would provide us is documentation or a recommendation by the FDA on how to use Cibo. It enables us to market Cibo under the agreed upon context as a drug development tool. And this would also allow the Cibo platform to be included in IND and NDA submissions without further review by CDER. So I'll just end the talk by saying that Cibo is indeed poised, for those that are interested in the audience, to evaluate your investigational agents in the clinic. We've worked out all the technical aspects of it, both preclinically and now clinically. Engineering and manufacturing is complete of all the devices, as well as our software package. We've already received regulatory clearance to perform our studies and guidance on how to perform studies with investigational agents. We have a network of clinics that are ready to work with us. So thank you very much.